And I'm honored to invite Bjorn Neumann from University of Cambridge and Amnon Buxbaum from Hebrew University on their talk about regenerative capacity of adult CNS progenitor cells. They're also Birax awardees with Robin Franklin, also from University of Cambridge.
Thank you. Can you hear me?
Yes, we can hear you. Thank you.
Okay, and the slide should be up, I hope, because I can. Yeah, I see it myself. Oh, great. Great. So to start, I would first like to start with my acknowledgements and really acknowledge Mikey Siegel and Kevin Chalut. Mikey was with me, a PhD student in Robin's lab. And throughout our time in Robin's lab, we worked on many different things that have to do with OPC aging. And Kevin was his co-supervisor and really drove the biophysical part of the data that I will show you. So the conceptual framework we work with is remyelination. And this is a spontaneous regenerative process that occurs in all vertebrates, CNS. So what happens is, for example, in diseases like MS, that myelinating oligodendrocytes can be destroyed, for example, by an attack of the immune system in this autoimmune disease. And then we have demyelinated axons in the CNS. Obviously, this leads to a loss of function because now we don't have saltatory conduction for which the myelin was there. But luckily, there is this endogenous regenerative process called remyelination. And during this process, oligodendrocyte progenitor cells can essentially, which are stem cells, they can enter the lesion site, then proliferate and then differentiate into oligodendrocytes again, and thereby restore not only saltatory function, but also ensure axonal integrity. But unfortunately, like all regenerative processes, also remyelination becomes slower with age. As a consequence, in aging, OPCs can still enter a lesion and they might still proliferate, but they usually do this at a speed that is so slow that they might not differentiate anymore in time to form new oligodendrocytes. And in this time frame before, the axon became vulnerable to degeneration. And because now we don't have an oligodendrocyte, it will essentially degenerate and then we lose neuronal function that we can't restore anymore by regeneration. So the question is then obviously, okay, what goes wrong with this aging process that inhibits the speed of remyelination? And what can we do about it? And quite simply, and what people like to say, it's always about like extrinsic factors that obviously lead to like intrinsic changes in a cell, and this leads somehow to a downward spiral that essentially results in this loss of regenerative capacity. But whatever the signal is, in the end, I think in the most simplistic view, we can say all of the signals integrate at the function of the stem cell itself and its ability to replace oligodendrocytes. And if we just look on the capacity of aged oligodendrocyte progenitor cells to proliferate, then we find compared to neonatal cells, they lose their ability to undergo proliferation. And even if we give them high doses of growth factor that normally drive proliferation, they can't really respond to it and they stay non-proliferative. Equally, if you take young OPCs and you put them into a culture dish and then you let them differentiate, normally they form beautiful MBP positive oligodendrocytes in a dish. If you do the same with aged cells, it just doesn't work. And if you give the aged cells then molecules that normally drive this differentiation process even faster, it also doesn't work in the aged cells. So this is really discouraging. And now you might ask yourself, okay, are these cells irreversibly broken? Because this is now a really important question if we want to do anything about it therapeutically. So to answer this question, we isolated neonatal OPCs and aged OPCs, and then transplanted them into neonatal brains, into a young environment. And if you do this and then see how well they proliferate and differentiate, then you will find no difference between the neonatal transplanted cells and the aged transplanted cells. So in a young environment, they function equally well, essentially letting us conclude that a young environment makes them always behave like young cells. Now, there are obviously many components to like a young environment that could account for that. But to cut a longer story a little bit short is that essentially, if you just take brain slices from young or aged animals and you decellularize them completely, so you're just left with ECM essentially, and then you reseed OPCs for example, from an aged or a young brain onto these ECM matrices, then you see, like in the transplant experiment, that if you place an aged OPC onto a neonatal matrix, then it starts proliferating again to neonatal levels, and it also starts differentiating again into oligodendrocytes, like in neonatal levels. I don't show you the converse experiment, but essentially the same thing would happen if you, in converse, would happen if you transplant neonatal cells onto an age matrix. They would just look age-like. So what about is it in the microenvironment that changes there? It could be, again, a lot of different things, like, for example, the composition of the matrix. But what we actually found, and which you can just feel essentially by just isolating the brains, is that the brain becomes stiffer when the older the animal gets. And this is actually like kind of like a linear process. So what does, in what form does actually the stiffening of the matrix contribute to the aging phenotype? So to elucidate that, we use hydrogels that developed in Kevin Chalhoub's lab, and we can tune the stiffness of the gels to soft or to stiff levels of like a neonatal brain or like an aged brain, or approximately. And if you place OPCs onto them, then what you see, if you take an aged OPC and you place it onto a soft matrix, then it proliferates and differentiates again to neonatal levels. While like if you put them onto a stiff substrate, then they don't do that, like they would do in an aged brain. And so this is quite a drastic phenotype in like a young brain, or just modulating the stiffness of a substrate. Obviously, we would try to understand like what is actually the molecular link between the changes in the physical environment during aging and the stem cell function. And you can think about a lot of things. Obviously, there is the extracellular matrix, and this is sensed by receptors on the cell. And then downstream of the receptors, there will be different signaling pathways that ultimately somehow integrate into the regulation of like, let's say, epigenetic function that then regulate whatever is the transcriptome, which determines then what is the phenotype of the cell, right? So we have different ways how we could interfere with this. We can, for example, interfere with the ECM by breaking down ECM, cross-linking with a chondroitinase, and this would, for example, work, and it would increase function, and we've shown this. Or we could do something about like how the cell perceives its environment, or how it integrates the signals intracellularly. An example, for example, would be blebbistatin, to just essentially avoid this like stiffening of the actin cytoskeleton, depolymerize it, and just essentially pretend to be in a soft environment. So we can do a lot of different things. And I would like to show you 2 examples. So the first thing I would like to show you is what happens if you delete the mechanosensor piezo1. Piezo1 is an ion channel, and the opening mechanism is essentially controlled by the stiffness of its environment. When it's stiff, piezo1 opens, and then there's an influx of ions, and it's not regulated or selective what ion goes in. So we thought, okay, if we now delete piezo1, our idea was, and it has something to do with stiffness sensing, then cells that have lost piezo1, they should just always think they're in a soft environment. So if we place them on stiff, they should always behave still like young cells. And this is exactly what we see. If we take aged OPCs that have way more piezo1, by the way, than neonatal cells, and we delete piezo1 from them, then we see that after piezo1 deletion, we see like culturing on a soft matrix, but now on a stiff substrate. These cells actually behave like neonatal cells. And the same is true for the differentiation capacity. So by just making OPCs not sense the stiffness anymore, we can convert them into cells that behave like younger cells. And then we can do a similar experiment and say like, okay, I haven't shown you the data in the first place, but if you take neonatal cells and you put them into an aged brain, they behave like aged cells. So we said, okay, if we delete now piezo1 from these neonatal cells and we would place them into an aged brain that is really stiff, would they sense it and behave like aged cells? So we did exactly that experiment. But again, once piezo1 is lost, the cells don't sense the stiffness of the environment anymore and behave like young cells. So I showed you something that is a receptor. And I showed you before in the diagram that obviously we try to understand how it gets from the ECM to the receptor to the integrating pathways to essentially, in the end, the nucleus, its lamina and the epigenetic integration. So what I would like to close with experimental data is like showing you data about like actually of the structure of the nuclear lamina in the aged cells and how it's regulated by the same processes and how modulation of it has the same effect as like a soft stiff environment. So to start with this, lamin B1 and lamin AC are differentially regulated in OPCs with age. With age, there's an increase in lamin A and a decrease in lamin B1 on transcript levels, on proteome levels. And we've shown this also by immunostaining in young or in aged brains. So, but then also these levels of lamin B and lamin C are essentially controlled again about if an environment is soft or stiff. So if you take neonatal progenitor cells and you place them on a soft environment, they stay essentially really high in lamin B1. They don't have lamin C like they would have in a neonatal brain. But if you place them on a stiff substrate, the levels of lamin B1 decrease, and at the same time, you see an increase in lamin C. And obviously, this correlates with the loss of function that the cells experience. So then we thought, okay, what happens if we just modulate then the ratio of A and B type lamins in the cells? And essentially, the experiment is to take then again OPCs out, transfect them with lamin B1, and then see what happens in the phenotype of proliferation and differentiation. So we took OPCs, transfected them, and again, like on a soft substrate or with piezo1 deletion, aged cells in which you overexpress lamin B1 start proliferating again, even in a stiff environment, and they also start differentiating again in a stiff environment. And I think if I just summarize like what I've shown you on the last like 15 slides or so, is really quite simple. Like the brain stiffens with aging, and a stiff environment makes a cell more like behave like an aged-like cell, a soft environment more like a neonate-like cell. And modulation of like any of the pathways that help with the integration of the signals or the sensing of it in the first place, let us phenocopy exactly this effect. Obviously, the really important question that we try to fix now is like trying to really get an insight, what are the effects of stiffness on the organization of the nucleus and its mechanics? And how do these changes in nuclear mechanics lead to the epigenetic changes and to this drastic phenotype change that we see? And with this, I would like to hand over to Amnon, who will tell you, I think, hopefully about some exciting stuff he's seen in nuclear mechanics and OPCs.